Greetings to all the viewers. I am N. Usha from 2nd MSc Microbiology, Sacred Heart College, Autonomous, Tirupattur. I am here to share about contamination, spoilage and preservation of milk and milk products. Contamination of milk. Contamination of milk, it occurs at two levels. First one is on farm and second one is during the transport and at processing. First on farm. Freshly drawn milk, it contains relatively few bacteria. However, Micrococcus and Streptococcus are usually found in aseptically drawn fresh milk. During the normal milking process, the milk is subjected to contamination from other region of the animals and other adjacent areas. Most commonly, the bacteria found in the manure, soil, and water contaminate or udder of animal from where they enter into the milk. Other possible source of contamination is hand and finger of milk or other dairy workers. Contaminations can also occur from dairy utensils. Second one is during the transport and at processing. During the transport and manufacturing process, the contamination occurs through the tanker, transfer pipes, sampling utensils and other equipments. Sometimes the pathogens may contaminate the milk from hand and finger of the milk handlers. This is a picture of contamination of milk. Microbial spoilage of milk and milk products. Milk is an excellent culture media for the growth of many microorganisms. So therefore, different types of microorganisms can grow in it and can cause the spoilage. Spoilage of milk. First one is souring. Evidence of souring of milk are sour flavor and then coagulation of milk to form solid like curd. Many lactic acid bacteria and other bacteria ferment the sugar of milk and produce the acids. Temperature of 10 to 37 degrees Celsius, Streptococcus lactis is most likely to cause the souring with possible growth of coliforms, enterococci and micrococcus. At a high temperature, 37 to 50 degrees Celsius, Streptococcus thermophilus and Streptococcus faecalis may produce a 1% low concentration of acids. Little souring occurs in milk held at refrigeration temperature. Pasteurization of milk, it kills more active acid forming bacteria but permit only the survival of thermoduric lactic acid bacteria such as the Streptococcus thermophilus and Lactobacillus, etc. Bacteria other than lactic acid bacteria may produce acids, specifically if conditions are unfavorable for the lactic acid bacteria. For example, coliforms produce acetic acid and formic acid. Similarly, the Clostridium can also produce butyric acid. We'll see the pictures in upcoming slides. Second one is gas production. It's also called strong fermentation of milk. Sugar fermenting organisms produce gas together with acid. Main gas form is coliforms, Clostridium, heterofermentative lactic, propionic bacillus, etc. Coliforms, Clostridium and bacillus may produce both hydrogen and carbon dioxide. Gas production in milk is evidenced by foam formation at the top of the liquid milk by gas bubble it trapped in the curd by formation of curd. Excessive gas production may cause the cracking or breakdown of curd is so-called stomy fermentation of milk. The Clostridium perfringens is mainly causes of stomy fermentation. Third one is proteolysis. The proteolysis is the enzymatic breakdown of milk proteins into smaller peptides. Changes is caused by proteolytic organisms include acid proteolysis in which acid production and proteolysis occur simultaneously, proteolysis with little acidity or even alkalinity, sweet curdling. Sweet curdling which is caused by renin-like enzymes of microorganisms. Acid proteolysis is caused by Micrococcus, Streptococcus faecalis where liquefacients and some lactose fermenting proteolytic Bacillus species. Sweet curdling is mainly caused by Bacillus series. So this is a picture of souring, gas production and sweet curdling. Fourth one is ropiness and sliminess. Ropiness of milk, it occurs both by bacterial and non-bacterial causes. First, non-bacterial ropiness, it occurs due to thickness of cream, film of casein or lactalbumin during the cooling. Bacterial ropiness is caused by slimy capsular material of bacteria which is usually developed at low temperature Bacterial tropiness is caused by slimy capsular material of bacteria which usually developed at low storage temperature. Bacteria producing the ropiness in milk are Alkaligenous viscolactis, Micrococcus frudenbrachi, Enterobacter aerogenus, Klebsiella oxytoca, and E. coli. The fifth one is changes in milk fat. Various bacteria, yeast, and mold hydrolyzes fat of milk and causes the rancidity. So, lipolytic organism species are Proteus, Pseudomonas fragi, Staphylococcus, Bacillus, Micrococcus, and Clostridium, etc. Pseudomonas fragi and Staphylococcus aureus may produce fairly heat-resistant lipase. Sixth one is alkali production. Pseudomonas fluorescens and alkaligen viscolactis may produce alkali. Alkali production is due to the formation of ammonia from urea and formation of carbonate from organic acids. The seventh one is flavor defect. It categorized into three types: acid flavor, caramel or burnt flavor, bitter flavor. First, acid flavor. The acid flavor may be aromatic or sharp. The sharp flavor is caused by production of acetic acid, formic acid, and butyric acid, etc. by coliforms and Clostridium. It is undesirable. Aromatic flavor is caused by Streptococcus lactis and Leuconostoc when they grow together. It is desirable. Next, caramel or burnt flavor. It is caused by Streptococcus lactic ware maltigens. Bitter flavor. It is caused by proteolytic organisms. Other flavor, they include earthy flavor by actinomycetes, fruity flavor by Pseudomonas fragi, and soapiness by Pseudomonas sapolactic, etc. The eighth one is color defect. The growth of pigmented bacteria and other organisms give undesirable color. Some examples include the four types of milk: blue milk, yellow milk, red milk, brown milk. Blue milk, it is caused by Pseudomonas cincinaneum. Yellow milk is caused by Pseudomonas cinchantha and also by Flavobacterium. Red milk is caused by Serratia mercens and Micrococcus roseus. Brown milk is caused by Pseudomonas putrefaciens and by enzymatic oxidation of tyrosine by Pseudomonas fluorescens. Spoilage of butter. Many spoilage microorganisms come in butter from cream or milk from which it is prepared. Color defect. Some color defect of butter is non-microbial. They include pink color is caused by sulfur dioxide refrigerant. Surface darkening is caused by evaporation of water from surfaces. For example, Strimphyllium gives black spots, Penicillium gives green spots, Alternaria or Phoma give brown spot, Pseudomonas nigrificans gives reddish brown spot, etc. Flavor defect. Cream and butter have capacity to absorb the moisture from surroundings. Some odors in butter is caused by growth of organisms include fishiness caused by Aromonas hydrophila, esters like flavor caused by Pseudomonas fragi, rancid odor caused by lipase producing organisms, yeasty flavor caused by yeast. Spoilage of cheese. Spoilage of cheese occurs either by mechanical damage or by microorganisms. Microbial spoilage of cheese occurs during the following three stages. First, spoilage during manufacturing. During the manufacturing of most cheeses, lactic starter culture is added to carry out lactic acid fermentation. Acid proteolytic bacteria may produce a bitter flavor. Leuconostoc may produce a holes in cheese. Various organisms causes the proteolysis, gas production, sliminess, and off flavor that damage the quality of cheese. Cheese with too low acidity because of failure of starter culture or because of addition of cream is often made slimy by alkaligenous, melalkaligenous, and Pseudomonas fragi. Spoilage during ripening. During the ripening process, the spoilage can occur by enzyme released from autolyzed bacteria and by the growth of microorganisms during the ripening. In most of the cases, like gas production by Clostridium, heterofermentative lactis, Propionibacterium, yeast, etc., is caused by eye formation or cracking of cheese. Clostridium also produce undesirable flavor by production of butyric acid. Certain lactic streptococci give the bitter flavor. Some bacteria and yeast give sweet, fruity, and yeasty flavor. In cheese with insufficient acidity, putrefaction is caused by anaerobic Clostridium. Microorganisms also cause discoloration on the surface of cheese. Oxidation of tyrosines by bacteria give the reddish brown to grayish brown color. Propionobacterium grow as yellow, pink or brown color complex. Spoilage of finished cheese. Soft cheese is most perishable and also hard cheese such as the cheddar and Swiss cheese are more stable. Most common spoilage organisms of finished cheese are molds. Some mold causing spoilage of cheese are Cladosporium black discoloration, Oosporia crustacea red spots, Penicillium puberulam green coloration, Monilea black disc. Next one is preservation of milk. Removal of microorganisms. Microorganisms from milk are difficult to remove. High-speed centrifugation called bacterification at 10,000 rpm. It removes about 99% of spores and more than 50% of vegetative cell plus some proteins. However, bacterification is not done commercially for removing the bacteria from milk. Mold can be removed physically from the surface of cheese by scraping or periodic washing. Use of heat. For milk, pasteurization is used for preservation. Pasteurization, it occurs two methods. First one is low temperature holding method. That is LTLT, low temperature for long time. 63 degrees Celsius for 30 minutes. High temperature for short time. HTST, it's 72 degrees Celsius for 15 seconds. Steam under pressure. 93 to 100 degrees Celsius or higher, it kills the spores. Sealed cans are evaporated milk processed 115 to 118 degrees Celsius for 14 to 18 minutes. UHT, ultra high temperature 138 degrees Celsius for 2 seconds. Use of low temperature. The refrigeration temperature is recommended for milk or similar products during the storage in plant, during the transportation, and during the storage in home until consumption. Fermented milk and cheese are chilled after their manufacture and kept chilled until they reach their consumption. Chilling storage is most commonly applied the technique of preservation of milk and milk products in home for short-term storage. Drying. Many types of milk products are prepared by drying or removing the moisture from them. There are two types of such products. One is condensed product and the other is dry product. One is condensed product is evaporated milk in which 60% of moisture is removed so that remaining high lactose concentration in solution is inhibitory to the microorganisms. Bulk condensed milk, sweetened condensed milk, semi-solid butter are other examples of the condensed products. Milk product prepared in dry form include dry milk, ice cream mix, etc. Milk can be dried by drum drying or spray drying. The final one is use of chemical preservative. There are two types of chemical preservatives, that is developed and added preservative, important in milk products. Developed acidity in many fermented milk products retard the growth of many microorganisms. Propionic acid developed in Swiss cheese is inhibitory to the mold. Sorbic acid, sugar is added to the sweetened condensed product. NaCl, that is sodium chloride, is used in the cheese manufacturing, gives the flavor and also act as a preservative. So these are all the overview about the contamination, spoilage and preservation of milk and milk products. Thank you.